internal environment or connective tissues, then muscle tissues, and nervous tissue. 4 types of tissues. And we start with the first one, with epithelial tissues. Epithelial tissues mm, can be located in different parts of our body. Epithelium of skin covers our body from outside. Also, epithelial tissues line all our hollow organs, which means organs with a cavity inside them, like esophagus and stomach. So they all are lined with epithelium from within, and um, also epithelial tissues make glands which produce some secrets. And also we have some specific epithelial tissues, very specialized to perform specialized functions, such as the function of um, hearing. So here in this picture you can see the picture of um, the internal part of the organ of hearing in our inner ear. Uh, this is the very important thing that you should know. So this is the classification of epithelia. You should know this diagram by heart. It is very important. So, first of all the epithelial tissues are divided into 2 large groups: surface or lining epithelia and glandular. Glandular epithelia make glands. Surface epithelia cover the body from outside and line the body cavities and hollow organs from inside. And surface epithelia are then subdivided into simple and stratified. Simple epithelia are subdivided into pseudostratified, squamous, cuboidal, and columnar, depending upon the shape of the cells and their location in these epithelia. And stratified are subdivided into keratinized, non-keratinized, and transitional. So we will discuss all the types of epithelial tissues now. All the epithelial tissues have specific features, which you can see here. This is also very important because these features can point to that you see exactly the epithelial tissue. For example, during the colloquium, uh, the professor can show you some tissue and you should decide is it an epithelium or not, according to specific features of this tissue. So, first, surface or lining epithelium is located on the border between the external and internal environment of the body. For example, like the epithelium of our skin, which is shown here, is located at the border between the external environment and internal environment of our body, connective tissue, blood vessels, etc. The same will be for the epithelia of intestine, for example, and stomach, because the content of intestines and stomach are actually the external environment for our body, because this content comes from outside. When we eat something, this something goes to our stomach, and this uh, portion of food may also contain bacteria, viruses, etc. So this stuff is external environment for our body, and epithelium, which lines our stomach from within, so locates between the external environment and internal environment. Next, cells form continuous layers using extracellular junctions. Like you see here, the epithelium tissue consists of cells. Here you can see cells um, with a lot of uh, extracellular junctions, junctions between the adjacent cells. You know that any tissue contains cells and extracellular matrix. But in epithelial tissues, we have almost no extracellular matrix, and all the cells are
آره.
um, located very close to each other. They have junctions between them and almost no extracellular matrix between them. The only extracellular matrix that we can see in epithelium is the so-called basement membrane, upon which the cells are situated. Basement membrane. So epithelium rests on the extracellular basement membrane that separates it from underlying connective tissue. So epithelium locates upon basement membrane, and below this basement membrane there there will be always loose fibrous connective tissue with blood vessels. So in this picture, this is loose fibrous
آره
connective
حالا
tissue, and this is the cross
اینم
section of
جالبه،
blood
حالا
vessel.
پایینش
The
اینم
same
مثلا
you can see
فایبر
here.
خیلی زیاد خودش رو
Um,
مثلا رد میکردید
the
دیگه.
basement membrane is located here,
حالا
and below
کارهای
it you can see
تنظیم
loose
دکترا و
connective
اینها.
tissue with blood vessels.
بچه‌ای که
Um, and these blood vessels give oxygen and nutrients to
اپیدورال خودش میره
epithelium.
دنیا.
These substances, like oxygen and nutrients, um,
وارد خودش نمیشه.
go from blood vessels to surrounding loose connective tissue first via diffusion process, and
چی؟
then
Walls of
also via diffusion
دیواره چی؟
they
نگاه کن.
pass through basement membrane towards the epithelial cells.
اوپنینگ
So
وایز
epithelium
آره دیگه اوپنینگ همون
gets
چیزان همون صورت.
um,
خیلی
oxygen and nutrition via
داشتم
diffusion
صحبت صحبت
from
می‌کردم.
underlying blood vessels in
تصویر
connective
صحبت
tissue.
کنیم البته که من نخونم.
The same you can see here.
آره
اگه
Uh,
شما بلد نباشید
in
اشکال
the
نداره.
right part, you can see just diagrams, and in the left part, two pictures. They are actually photographs taken from microscope. So here you can see epithelium lying upon basement membrane. Basement membrane um, doesn't stain with any um, dyes. So you cannot see basement membrane, you just can see the border
تصویر
between the
چهار.
epithelium and underlying connective tissue. So the basement membrane locates just at this border, and
خوبه
below
دوشنبه
it you can see
مونتا
connective tissue with
ساعت
cell nuclei of cells of connective tissue
12 آنلاینی، ساعت 12
این
و نیم.
آنلاینه؟
and
نه
stained
اون چیزش
دوشنبه
eosinophilic.
تا ساعت 12
And
آنلاینه؟
here cross sections
واقعا؟
of small blood
این
vessels
یکی
of
داره؟
چرا
capillaries
ولی
through
نوشیدنی
which the
اینجوری
epithelium will get
آها.
20
its
ساعت روزی.
um,
کدوم
nutrition and
ولی
oxygen. And the same is
هفت
here.
میری؟
Basement
چرا
membrane
نمی‌دونی؟
locates here,
استوری
and
چی؟
below it,
لیکت
connective tissue with blood
آنلاینه؟
vessels.
تو باید
اونم
فیلم می‌ذاره.
آنلاینه. نه آنلاینه.
Next feature of epithelial
آنلاین
tissue
خیلی عقب.
is that the cells have marked apical basal polarity
میشه لکچر
and
لکچر
distinct,
نیست
distinct apical lateral and basal
یعنی
plasma membrane domains. It means
اشکری
that
نداره.
if you
حالا
look
مثلا
at the
یادداری
cell of
بدم
the epithelial
یادم
tissue,
نمی‌دی.
you can say where is its top surface and where is its bottom surface because they look
واقعا نمی‌دونم
different.
چی میشه شروع
You
کنم.
can easily say that this is the top, the apical surface, because it has some specific specializations of the apical
15 تا
surface,
شده
like
باید باشه.
microvilli shown here,
نهایت
and
10
some
خب چند تا بچه
basal
داری؟
labyrinth
17.
with basal foldings on the basal surface.
ماری جلو.
And on the lateral surfaces, we will see the junctions between the cells. So the same is here. You can easily say that
انگلیسی چقدره
here is
حضوریش؟
the apical surface and here is basal
دانشگاهی؟
surface because here the cells stay upon the basement membrane.
دانشگاهی؟
Where the basement membrane is, there
بچه
the
چند تا کلاس
basal
هم هستی
surface
کلاس
of
داری؟
the epithelial tissue is located. And
بعد تا کلاس هم
on the
هستم
apical
دیگه.
surface here we can see cilia.
چی میگه؟
کی
میگه
This
یه
is
ساعتی
for
وایسا.
چی اصلا
simple
کنترل تو نمیکنه
epithelium.
همه چی
It
میکنه
means
میشه.
simple
چند تا
means
کلاس هم
that
میشه؟
it has only one
نه یک
layer of cells. All the cells stay upon basement membrane. But here we can see the stratified epithelium. It consists of several layers of cells, and only the first layer, more bluish here in this picture, actually locates upon basement membrane. And um, what means apical basal polarity for stratified epithelia? It means that um, you can easily say where the basal layer is because it locates upon basement membrane, and below it the connective tissue will be located. And if you go from basement membrane towards the surface of such epithelium, you will see the cells of this epithelium at different stages of their life cycle. So in basal layer, we will always have stem cells, Campbell cells, which divide and make new cells. And then when we go upwards, we will see the mature cells, then aging and dying cells. Here you can see the so-called desquamation of the surface epithelial dead cells from the surface of the stratified epithelium. So both simple epithelia and stratified epithelia have so-called apical basal polarity. So it means that you can say where is the top and where is the bottom in this tissue. If you compare epithelium to connective tissue, for connective tissue you cannot say where is the bottom, where is the top, because the cells have no polarity. They look the same from all the sides. So for connective tissue, no apical basal polarity is seen. And finally, the last feature of epithelial tissue is that they have high regenerative capacity. The most of the epithelia are the renewing cell population. It means that we have the constantly dividing stem cells located always closely to the basement membrane, and all other components of differon, including mature cells, aging and dying cells. And dying cells are replaced by newly produced cells from the Campbell cells. So the cellular composition of renewing population include these stages: stem cells, dividing cells, located in basal layer for stratified epithelia, or in different areas for simple epithelia. So this is the pseudostratified epithelium, and here stem cells are located also closely to the basement membrane. This is the epithelium of trachea actually. And in these two pictures below, you can see the epithelia of stomach in left part, gastric epithelium, and in right part, the intestinal epithelium. They both are simple epithelia because the cells lie in one layer. All the cells lie upon basement membrane, and here also. And these epithelia have stem cells in specific places. So in stomach epithelium, the stem cells are located always here. This invagination is called gastric pit, and these are gastric glands. And where the gland goes to gastric pit, there the stem cells are located. You shouldn't learn this information now. You should just know that in different epithelia, stem cells can be located in different places. For simple epithelia. For stratified epithelia, where we have several layers of cells, stem cells are always located in the basal layer, which lies upon basement membrane. In intestinal epithelia, stem cells are located closely to the bottom of the so-called crypt. So this is the crypt, this is the villus, and the stem cells are located in the layer of epithelium inside the crypt. So renewing cell population means that natural decline in the cell number is balanced by proliferation of stem or Campbell cells. Now, cell surface specializations, you know them from the previous topic. So you can find cilia or microvilli or stereocilia or kinocilia upon the apical surface of some cell cells, not upon all of them, but upon some of them only, basing upon the function of the epithelium and the function of the cells particularly. Here you can see the functions of such of the cell surface specializations, microvilli and cilia. Um, Between the cells in epithelium we have the cell junctions, and in the basal layer, um, if our cell is located upon the basement membrane, it will always have the hemidesmosome for attachment of the cell to the basement membrane. And some of the cells that perform the function of active transport of ions against the concentration gradient, such cells can have the basal labyrinth with folds of membrane and mitochondria between those folds. So, types of epithelia tissue. All the epithelia are divided into two large groups: surface or lining epithelia and glandular epithelia. So, surface or lining make one or several layers of cells located upon basement membrane, and they separate the internal environment of our body, which is below the basement membrane of this epithelium, and extra um, outer environment of our body, such as outer environment, the air outside of our skin, or the content of our stomach and intestines. So this is the surface epithelium. And glandular epithelium consists of cells also lying upon basement membrane, and these are glandular cells. Their function is to produce some substances such as proteins, lipids, steroids, mucus, etc. And we can find both types of epithelia in some organs. For example, this is the part of the cross-section of the esophagus. Esophagus leads from our pharynx to stomach. So this is the tube which leads to stomach. This is the lumen. Lumen means the internal cavity of the hollow organ. So this is the lumen. The food will be here. And this is the wall of esophagus. And the cavity, which is called lumen, is lined with surface epithelium, which is here. This is surface epithelium. And inside the wall we can find the glandular epithelium. In the case of esophagus, here we will see the glands which produce mucus. Mucus serves for moisturizing oxygen when the food goes from our mouth towards stomach, to decrease friction, um, to prevent damage of epithelium caused by hard particles of food. So mucus produced here and released via ducts to the surface of epithelium. So the epithelium of our esophagus is always covered with layer of mucus produced by glandular epithelium. And now we start to discuss all the types of epithelia one by one, and we will start with surface simple epithelia. So types of surface epithelia include simple epithelia, which are in left part, and stratified epithelia. In simple epithelia, all cells lie upon basement membrane. All cells, here you can see in each picture, all cells have attachment to basement membrane. In stratified epithelia, only basal layer of cells uh, lies upon basement membrane. So these cells of basal layer like have contact with basement membrane, and superficial layers have the contacts not with basement membrane but with underlying layers of cells. So these cells in this epithelium have junctions only between the adjacent cells. And now about the classification of these epithelia. So, simple epithelia are subdivided into columnar, cuboidal, squamous, and pseudostratified epithelia according to the shape of the cells. So, a simple columnar epithelium contains high elongated cells, which are kind of standing upon the basement membrane. So they are elongated and they have elongated nuclei, which are oval and kind of standing. Columnar means the shape of these cells, which are like columns. Another name for this epithelium is prismatic, because in 3D, three-dimensional picture, these cells are like prisms. Next, simple cuboidal epithelium, as you can see from this name, have the cuboidal cells. They are square in the two-dimensional picture, and these cells have round cell nuclei. Simple squamous epithelium contains flattened or squamous cells, which are kind of lying upon basement membrane, and they have squeezed, flattened cell nuclei. Actually, we usually, when we look at the real pictures from the light microscopy um, pictures, we cannot see the borders between the cells. And then we should look up to the shape of cell nuclei. And if we see that cell nuclei are oval, we can say that epithelium is simple columnar. If we see that cell nuclei are round, then we can say that epithelium is cuboidal. And if we see that nuclei are flattened, then we can say that epithelium is squamous. We will see it later in pictures. And the last type of simple epithelium, it is called simple pseudostratified epithelium. Pseudo means, this prefix means looks like but not really is. And this word pseudostratified means that this epithelium looks like it is stratified. Stratified is here, for example. So this looks like stratified, but not really is stratified. Why? Because here all the cells have the contact with basement membrane. But in real stratified epithelium, only cells of the basal layer have junction with basement membrane, and other cell layers have junctions only with adjacent cells. So simple stratified epithelium, all cells have junctions with basement membrane, but their nuclei are located at different levels. And it is because these cells actually have different height, because they are the cells of the differon at different stages of life cycle. Those which are shorter and closer to basement membrane, they are stem cells. Those which are somewhat higher, they are the cells in the process of differentiation, and the highest cells which reach the surface of this epithelium, they are the mature cells, fully differentiated cells. Next, in stratified epithelia, only basal layer of cells lies upon basement membrane, and here we have three types of stratified epithelia. First one, stratified squamous non-keratinized. Next, stratified squamous keratinized. And stratified transitional epithelium. Actually, you should learn all the words in each name of epithelium. So, if you are asked about the type of epithelium, you should name all the words in this sequence, like stratified squamous non-keratinized, because all these words have their meaning. So this is stratified because of the layers, and only basal layer lies upon basement membrane. Next word, squamous, means that the cells of the superficial layer, which are located at the top of this epithelium, are squamous, like in simple squamous epithelium. They are flattened. And then non-keratinized means that upon the surface we have living cells. They are not keratinized. They are not like dead squams in the skin epithelium, and not like not as hard as hair or nails, which are keratinized structures. So here we have non-keratinized epithelium because at the surface we have living cells. Next, stratified squamous keratinized. Unlike non-keratinized, if we look at the surface layer, we have here dead cells or squames. How we can say that these are dead cells and these are living cells? Because of the presence of or absence of cell nucleus. Dead cells have no cell nucleus. So we can easily say that this is keratinized epithelium because of dead cells layer. And finally, transitional epithelium or urothelium. This is specific epithelium which lines the um, urinary bladder, for example. And this epithelium changes its shape somewhat in different mm, degree of stretching of the urinary bladder. So if the urinary bladder is filled with urine, then it stretches, it becomes larger. And what happens to epithelium? Epithelium becomes stretched, and it has fewer number of layer of cells, just 1 to 3, uh, thinner wall, so the thickness of epithelium is decreased, and the cells of the superficial layer become flattened, but their cell nuclei are still round in shape. When the epithelium is not stretched, for example, if the urinary bladder is not filled with urine, then the thickness of epithelium increases, so it is more thick here. Number of layers increases, so here we have 1, 2, 3, 4, 5 layers of cell nuclei, and the shape of the superficial layer of cells changes from flattened to more rounded. So this is why it is called transitional, because its shape changes depending upon the rate, rate of stretching of this epithelium. So here you can see how these epithelia really look like. So in the first picture we can see simple squamous epithelium. It is simple, one layer of cells lying upon basement membrane, and the cells are flattened. So inside view, if we make the section of this epithelium, what we will see? We will see the flattened cell nuclei, which are located here. And below them, this is connective tissue, below this layer of epithelium. If we look at this epithelium from the top, here is the top view of the same epithelium, we will see the shape of the cells. So here you can see border between the cells, and you can see that the cells have a regular shape. Each cell has its nucleus inside it. Next, a simple cuboidal epithelium. So it looks like this. One layer of cuboidal cells, which are square in shape with rounded cell nuclei. So you can see it here. The basement membrane is located here. And these are the cells. They are cuboidal in shape and they have rounded cell nuclei. Next, simple columnar epithelium. One layer of cells. cells are columnar, elongated, with elongated cell nuclei. So here is this epithelium. Basement membrane locates here. Below it we will see loose connective tissue, and these are the cells of this epithelium. We not always can see the borders between each cell, but we can see the shape of cell nuclei. Cell nuclei are elongated. And finally, simple pseudostratified epithelium, where all the cells have contact with basement membrane, but their nuclei locate at different levels. So this is it. This is the epithelium of trachea. Basement membrane is here, below it connective tissue, and cells are columnar, elongated, and their cell nuclei locate at different levels. Stratified epithelia are shown in this picture. In top two pictures you can see transitional epithelium. In left picture it is not stretched, in right picture it is stretched. So in left picture the number of rows of cell nuclei is higher than in the right picture. And if we look at the shape of the cells of superficial layer, in the left picture where the epithelium is not stretched, the shape of the cells is rounded, or it is called dome shaped. And here the cells are more flattened in the superficial layer. Next, stratified squamous non-keratinized epithelium in the left bottom picture. Stratified epithelium because it has several layers. Squamous because the cells in the superficial layer have flattened cell nuclei. We cannot see here the borders between the cells, so we cannot say what's the shape of the cells is here, but we can see the shape of their nuclei. Their nuclei are flattened. It means that the cells are flattened, so we can say that this is the squamous stratified epithelium, and it is non-keratinized because at the superficial layer we can still see the cell nuclei of the cells. It means that here we have living cells. They are alive because they have cell nuclei. And in the last picture we can see stratified squamous keratinized epithelium. Basal layer is here, below it connective tissue, other layers of cells, and here is the last time when we see the cell nuclei in cells. And all this is the keratinized layer, which contains non-living cells, dead cells, cells squames. So it is keratinized epithelium, stratified squamous keratinized epithelium of the skin. Now, here are the embryonic sources of development of different types of surface epithelia. This picture shows that all the germ layers give the epithelia. Ectoderm gives epithelia. Ectoderm, the outermost layer of the embryo, it gives stratified squamous epithelium of skin, epidermis, also epithelium of cornea in the eye, epithelium of the oral cavity vestibule, and epithelium of salivary, sweat, sebaceous, and mammary glands. So they are from ectoderm. Mesoderm gives also the epithelia. So epithelia of renal calyces, renal pelvises, ureter, epithelia of seminiferous tubules and sperm ducts. It means the epithelia of urinary and reproductive system. Renal, it is about kidneys. Seminiferous tubules, sperm ducts, these are the organs of male reproductive system. And epithelial of uterus and fallopian tubes, these are organs of female reproductive system. So nephrogonotome, which is the part of mesoderm, gives us the epithelium of urinary and reproductive system. These epithelia are listed here. Endoderm give us the epithelium of intestine, stomach and its glands, liver and pancreas, and also the gallbladder and bile ducts. And also neural tube and neural crest can give epithelia, etc. Precorder plate gives us the epithelium of trachea and bronchi, and also epithelium of esophagus. So this is actually what you should know. And now we start to look at the particular types of epithelia. Our first epithelium type is the simple squamous epithelium, which has its specific name. It is called mesothelium. Mesothelium. This is the picture of histological preparation. It is called whole mount mesenteric preparation. If you remember the types of histological preparations, we can have the total preparation or whole mount preparation, which is not a section. It's a rather thin film containing such a tissue, which is stretched upon the glass for microscopy, stained, and this is what we can see. So this will be the surface type of epithelium. So this is our classification of epithelium. Surface, simple squamous epithelium. This is how it looks like if we make a section of such epithelium. But this is not a section, this will be the top view of this epithelium. Next, what is mesenteric preparation? And what is mesothelium? Mesentery, it is shown here. It is the film through which blood vessels go, which give blood supply to intestines. So mesentery kind of helps the intestine to be attached to the wall of the abdominal cavity, and also through the mesentery blood vessels and nerves go to intestines. Mesothelium lines the body cavities, abdominal cavity, which is shown here, inside which the intestines are located, pleural cavity where lungs are located, pericardial cavity where the heart is located. So all these cavities are lined with mesothelium, and also mesothelium covers all the organs in these cavities. So the intestines are also covered with this epithelium, mesothelium. Lungs and heart, which are located in pleural and pericardial cavities, are also covered with mesothelium. And mesothelium provides free movement of these organs inside cavities and prevents their adhesion and friction to the cavity wall, because mesothelium produces serous fluid which fills these body cavities. So the wall of abdominal cavity is covered with mesothelium, and the intestines are covered with mesothelium, and between these layers of mesothelium there is the serous fluid, which decreases friction when the intestines move inside the body cavity, because during the digestion process the intestines can a little bit move inside the cavity. And the same for heart. When heart beats, it moves inside the pericardial cavity. And mesothelium helps to decrease friction because it helps to produce serous fluid which fills those cavities. So, embryonic sources of development of this epithelium. Embryonic source of development of the mesentery is the visceral layer of splangnotome. So this is the mesoderm. It has somite, nephrogonotome, and lateral plate mesoderm with the parietal layer and visceral layer. The visceral layer of splangnotome will make the mesentery, which cover the organs inside body cavities. The mesothelium from parietal layer will make the mesothelium which lines the cavities. So, what happens with embryo? In the picture below, we can see that the lateral folding happens, and now we should look
What you
at,
see from
at
video?
these visceral layers of splangnotome.
Haydi
What will
güzel.
happen to them?
Güzelmiş.
The
Nasıl bakayım?
endoderm will close and make the gut. Now it is shown with yellow color here. And what happens to the layers of splangnotome, visceral and parietal? As a result of the lateral folding, these layers fuse together, visceral with visceral, parietal with parietal, and this is what we see now. The visceral layer of splangnotome is here, and parietal layer of splangnotome is here. What will be between these layers? Between these layers will be the body cavity, the abdominal cavity, inside which the intestine will be located. And this will be the mesothelium, which covers the intestine. It is from visceral layer of splangnotome. And this will be mesothelium, which lines the abdominal cavity from inside. And this develops from parietal layer of splangnotome. And between them, we will have the abdominal cavity filled with serous fluid. And here, next stage of the same process. So this red line here shows the mesothelium. This is the intestine, its gut. Its epithelium is yellow in this picture. It is covered with mesothelium from visceral layer of splangnotome. Here is the body cavity, abdominal cavity, lined with mesothelium from parietal layer of splangnotome. And as you can see here, the um, visceral layer of splangnotome from left and right sides fuse together, as you can see here, and elongates. And this area where the left and right part um, comes close together and elongates, this area will make the mesentery, which is finally is shown here, and it looks like this. So the intestine actually is kind of hanging on this mesentery. So the gut hangs on the mesentery, which is actually the two layers of the mesothelium from right and left side. And this is actually what we see in this histological preparation. So our histological preparation is the part of this mesentery, of this area, upon which our intestine hangs inside our abdominal cavity. So this is how it really looks like. This is the silver impregnation staining and hematoxylin. Hematoxylin was used to visualize the cell nuclei, which are rounded in shape and they are more purple. And silver impregnation makes the cell boundaries visible for us. So we can see here the borders of the cells. These holes are the artifacts. Artifact is the damage caused to tissue as a result of the action of the scientist. So it is not the breaking down of the tissue inside the organism, but it is the result of the histological preparation. At higher magnification, we can see the cells with their cell borders, cell 1, cell 2, between them the border, and here we will have the cell junctions between these cells. We cannot see these junctions in light microscopy, but we know that they exist because the cells are of epithelial tissue and they should have cell junctions between them, including tight junctions and desmosomes. Now, if we look back here, we can see that we see, for example, one cell here and the borders of another cell below it. It is actually because here we see not only one layer of cells, but two layers attached together. So once again, what we see? We see mesentery, which represents two layers of mesothelium from left part and right part, which come close together. How it will look like you see here, the mesothelium, it is simple epithelium, squamous with flattened cells, with cell nuclei here, borders between the cells. The cells lie upon basement membrane, and we have two layers of this epithelium. One layer here, one layer here. And between those layers, we have loose connective tissue. Both layers of mesothelium lie upon basement membrane, which is brownish in this picture, and always below the basement membrane in every epithelium we will have loose fibrous connective tissue. This is the picture of how this epithelium looks like if we make the cross section. So these are the cells of mesothelium, one layer here, one layer here, and loose fibrous connective tissue with blood vessels between them. As you remember, mesentery gives blood supply to intestines. So between these layers of mesothelium, the blood vessels go towards intestines. So we see here also blood vessels. And this is why we see here two layers of cells. So cell borders, cell nuclei. Some of the cells have two nuclei. This is because of the incomplete cell cytoplasm division during mitotic cycle. In your album, you should draw two pictures, the top view of this preparation, where you should draw something resembling this picture, with the cells of irregular shape located closely together. Their cell borders are black because of silver impregnation staining, and cell nuclei inside the cells. And in picture B, you should draw the side view as a result of cross-section of this epithelium. If we make the cross-section like this is what we will see. Simple squamous epithelium lying upon basement membrane and loose fibrous connective tissue below it. So about all the epithelia that we discuss, you should know their structure, location, the name, full name of the cells, type of population. This is the renewing population. All of the epithelia that we study are renewing population, and embryonic source of development. So for mesothelium, the name of epithelium is mesothelium. According classification, it's the simple squamous epithelium. The name of the cell is the mesotheliocyte. Mesotheliocyte. Mm, Next, the embryonic source of development is the lateral plate mesoderm, the visceral layer of splangnotome, and also the location in our body. So location of our body is the mesentery. So it's the mm, mesothelium which covers the organs in body cavities and which lines the body cavities from inside. Also, we will see the mesothelium in our other histological preparation, in the preparation of the section of small intestine. So this is the section of small intestine. Intestine is here. It is the hollow organ with the cavity, which is called lumen inside. In lumen, the food will be during digestion process. The embryonic endoderm, which is yellow here, will become epithelium, which lines the intestine from inside. The lumen will be here. And the intestine is covered from outside with mesothelium, red line here. So this is the section of the wall of intestine. Lumen is here, internal surface is here, and here is the external surface, outer surface, and it is covered with mesothelium, which will look like this. At high magnification, this is how this area looks like. So you can see here flattened cell nuclei. We cannot see here the borders of cells, just the cell nuclei. These cells are located upon basement membrane, which is not seen, with a thin layer of connective tissue below it, and then the smooth muscle tissue goes next. So all of this is smooth muscle. Next we go to this preparation section of small intestine, and we look at the simple columnar epithelium with brush border, which covers these finger-like protrusions, evaginations of mucosa of the intestine, which are called villi. So our small intestine locates in abdominal cavity. It has inside it folds. So this is fold. These are villi. Sorry. Villi, these protrusions, and every villus is covered with epithelium. And epithelial cells have microvilli. Microvilli we studied last time when we studied cell surface specializations. So microvilli can be seen only in electron microscopy picture. They are located at the top surface of the epithelial cells. So in our histological preparations, we cannot see microvilli, and what we see here, these are just villi. So don't mistake villi with microvilli. Villi are large structures which are easily seen in light microscope, but microvilli are small structures located at the top surface of each cell of epithelium, and they are seen only in electron microscope. Why a small intestine has folds, villi, and microvilli? The answer is to increase surface of the internal surface of the intestine, to increase rate of digestion and absorption process. So intestine is for digestion and absorption, and it needs to increase the surface area. So we see here columnar, simple epithelium with brush border. It is columnar because the cells are elongated with elongated cell nuclei. So this is the type of surface epithelium, surface simple columnar epithelium. Embryonic source of development is embryonic endoderm, which is shown here with green color. Where is it? Where is this epithelium? It is covering all the villi from outside here, and also it goes here. These smaller invaginations which go deeper, these are called crypts. So this epithelium covers both evaginations, villi, and invaginations, crypts. the simple columnar epithelium with brush border. What means brush border? As you remember, this is how microvilli look like when we look at them in light microscope. So this is the line covering the apical surface of this epithelium. In this epithelium, we have two difference of cells, or what does it mean? It means that we have here two cell types. First cell type is the columnar epithelium cell with microvilli. So most of the cells here, number one, they are epithelial cells with microvilli or with brush border. Um, their function is to produce enzymes for the last stage of the digestion process, for breaking down of dimers to monomers, and their subsequent absorption. Absorption means that those monomers, like amino acids and monosugars, go from the lumen of intestine through the cell membrane of microvilli into the cytoplasm of these cells. And then these molecules go through the cytoplasm of the cells, then they go out from the cells through their basal surface, then through basement membrane, into the underlying connective tissue, and then into blood capillaries with the help of diffusion process. And this is how we ingest our food. And second cell type here is called goblet cells. Goblet is the name of the cup for drinking, and this describes the specific shape of these cells. Goblet cells number 2. They have in the standing part of the cell, and most of the cell is filled with granules with mucus secrete. So these are granules with mucus, and when it is needed, mucus is released outside. And this process we can see here. The granules are released by the cell. So at high magnification, how this epithelium looks like. So first of all, where is the border of epithelium? It should be here. So here is the location of the basement membrane of this tissue. So basement membrane goes like this, and this is the row of the cell nuclei of this epithelium. So this simple columnar epithelium with brush border covers the surface of villi. One villus here, second villus here. They both are covered with this epithelium. It is simple because all the cell nuclei are located at one layer. They make one layer of cells. It is columnar because of the shape of cell nuclei. We cannot see here actually the borders between the cells, but we can see the shape of their nuclei. Their nuclei are elongated, so we can say that the shape of the cells is also elongated or columnar. So this is columnar epithelium. Brush border is this thin line covering the apical surface of these cells. In the layer of this epithelium, we can see white bubbles, and these white bubbles are actually the cytoplasm of goblet cells filled with mucus. Mucus is not stained with eosin or hematoxylin, and it stays unstained. And these are cell nuclei of goblet cells. At high magnification, the same, the epithelium. Here will be the basement membrane. It is not seen, but here the epithelium ends and loose fibrous connective tissue begins. At the top surface of this epithelium, we have brush border, the elongated cells with elongated nuclei. They are the columnar cells with brush border. And here we have the goblet cell with mucus. So at electron microscopy level, we see here both cell types. the columnar epithelial site with microvilli, and here also one of them here and second here, microvilli. Um, this is the terminal web inside the cytoplasm of the cell, to which the actin microfilaments of microvilli are connected. The cytoplasm is filled with different organelles such as mitochondria, transport vesicles. Golgi complex should be above the nucleus, and some mitochondria and rough endoplasmic reticulum. Rough endoplasmic reticulum here is to produce the enzymes which will make the last stage of the breaking down of food molecules. And second type of cells is goblet cell. Goblet cell is here and here. Nucleus is located in its basal part. The cytoplasm around the nucleus is filled with rough endoplasmic reticulum. Above the nucleus, the Golgi complex should be located. It is not seen, but it should be here because we know the function of this cell. The cell is mucus-producing cell, so we know that it should have rough endoplasmic reticulum and Golgi complex, because mucus is the mixture of mucins. Mucins are molecules which have two components, protein and sugar. Protein component of this molecule is made up inside rough endoplasmic reticulum, and then sugar component is made and added to the protein part inside Golgi complex. And Golgi complex also makes vesicles with this content, and these vesicles fill most of the cell. So here are the secretory granules or vesicles filled with mucus, ready to be excreted from the cell when it is needed. Another picture of columnar epithelium cell with microvilli. So here is its apical surface with microvilli. Microvilli in cross section are shown here. Lateral surfaces are seen here. So going downwards from the top surface, first junction will be tight junction, then should be intermediate junction or belt desmosome, and all other types of junctions will be below. For example, these are interdigitations, and here also. And this is the basal surface of the cell. Here, this grayish line, this is the basement membrane upon which this cell is situated. So it should have here hemidesmosomes for attachment to basement membrane. What is below? Below is connective tissue with blood vessels. Blood vessels are not seen. We see here just some components of loose fibrous connective tissue. Here you can see the top part of 3 cells of this epithelium. So why 3? Because here in the right part we can see the part of cell 1, and here is the area of cell junctions between cell 2 and cell 3. So in this picture we can see actually parts of 3 cells. Apical surface of these cells has microvilli covered with glycocalyx. Glycocalyx here serves not only for reception but also for digestion because of the enzymes connected to the cell membrane. And cell junctions include tight junction, which is always first, belt desmosome at the level of terminal web, which is grayish, it is here, and next spot desmosome. Another example of cell junctions between columnar epithelium cells with brush border. So first one is tight junction, also seen in the right picture. Next will be ZA is for zonular adherens. This is the Latin for belt desmosome. So it is always located at the level of terminal web, which is grayish. It contains microfilaments. It is here, belt desmosome. Next, here we can see spot desmosome. not seen in the right picture. In the right picture, we see instead interdigitations. Um, Two electronograms of goblet cells and the light microscopy picture of this cell. So nucleus is triangular in shape in its lower part. It is surrounded with a lot of rough endoplasmic reticulum, which gives the basophilic staining of cytoplasm. You know that rough endoplasmic reticulum makes basophilic staining, and most of the cell is filled with non-stained granules with mucus. So in your task 27, you should draw such epithelium of intestine, which includes two cell types, the um, cells with microvilli and a goblet cell. Um, and also, what should you know? Where the stem cells are located. Once again, we should go here and look at the diagram in the bottom right. So here you can see the intestinal epithelium diagram. Uh, villi, intestinal villi are here, and between them below you should see here the crypt. This is called the crypt. It is lined with the same epithelium, and the stem cells are located in this epithelium in the crypt. Mitosis points to stem cells. Stem cells divide, and mitosis can show us where the stem cells are located. So for intestinal epithelium, stem cells are located in particular area in crypt. Where the crypts are in our histological preparation, once again, crypts are these. So these invaginations of epithelium are crypts, and the stem cells will locate here in epithelium lining crypts. And while the cells mature and become specialized, they move along basement membrane towards the top of villus, and then they age and die, and aged dead cells are shed away from the top of each villus, and they are constantly replaced by newly produced cells, moving along basement membrane from the crypts. Embryonic source of development for this epithelium is endoderm, location inside intestine, function, you know. Two types, two cell types here, and it means the two differons, and the stem cells are the stem cells for both differon. So the stem cell can develop either into goblet cell or into the columnar cell with brush border. Okay, go next to the ciliated pseudostratified epithelium in the section of trachea. So this is trachea. Trachea leads to lungs. This is for breathing. And here is the part of its cross section. So this will be the lumen, the internal space of trachea, and trachea is lined with epithelium from within. So this will be the epithelium of trachea. Embryonic source of development of this epithelium is precaudal plate, which originates as a result of migration of the cells through the Hansen's node during the second stage of gastrulation, immigration. So this is the Hansen's node, and the first mass of migrating cells make precaudal plate, and the cells which go next make the notochord here. So we need this area. In this picture, the precaudal plate is here, and this will be the notochord. So precaudal plate makes the ciliated pseudostratified epithelium of trachea. Another picture of the same preparation. So the lumen of trachea is here. The epithelium which we need, it is located here. So a ciliated pseudostratified columnar epithelium. It is simple. It means that all the cells have contact with basement membrane. Pseudostratified means that it looks like stratified but not really is, because all the cells contact with basement membrane, but their cell nuclei locate at different levels. Why so? Because here we have the cells at different stages of development. The lowest row of cell nuclei, the nuclei which are located the most closely to the basement membrane should be here, at the border of epithelium and underlying loose connective tissue. So the lowest row of cell nuclei is the row of basal cells, or Campbell or stem cells, which function is to divide and make new cells. So these are basal cells. Next, the second row of cells, number 2 in this picture, these are called tall intercalated cells. Intercalated means inserted, kind of inserted between mature cells. So they are a little bit higher, like this or like this. These are the cells in the process of differentiation. So they are not stem cells, they cannot divide anymore, but they are still not mature cells. They are in process of differentiation. And finally, the most top row of the cell nuclei in this epithelium is the row containing two types of mature cells, of two different ciliated cells and goblet cells. Goblet cells are almost the same as the goblet cells in intestinal cells. They look the same. They um, their function is the same, also to produce mucus and release it to the surface. And what about ciliated cells? Ciliated cells have cilia upon their surface, and so here in this epithelium we have two cell types, or two differons of ciliated cells and goblet cells. And the function of this epithelium is called mucociliary transport, or the same mucociliary clearance. Clearance from to clear, to clean. What does it mean? It means that um, mucus produced by goblet cells covers the surface of this epithelium, and dust particles and bacteria, which you can see here, stick to this sticky layer of mucus, and then the beating of cilia moves this mucus along the trachea uh, from lungs upwards towards the pharynx, where this mucus with bacteria and viruses and dust particles is swallowed or coughed away from our body. So here we have um, four kinds of cells, but only two cell difference. Um, why so? Because basal cells and tall intercalated cells are actually the cells of these two differons. Basal cells are stem cells for both differons, and tall intercalated cells are cells in the process of differentiation, either into goblet cells or into ciliated cells. In electron microscopy pictures, this is how such epithelium can look like. So here we can see the ciliated cells with cilia upon their apical surface, and these are basal cells, which are stem cells. Stem cells are undifferentiated cells, and as you remember, they have high nuclear to cytoplasm ratio. It means that they have very few cytoplasm if we compare these cells to the mature cells. Mature cells have a lot of cytoplasm because they should have a lot of organelles in cytoplasm to perform the function of such a cell. The function of these cells with cilia is to um, move the fluid with the help of cilia, and this movement needs energy in ATP form, and the ATP is produced by mitochondria. So here we can see a lot of mitochondria in the apical surface, in the apical part of the cell. Another picture of this epithelium. The basement membrane is somewhere here. So here's the border of this epithelium. Next, the basal cells, which cell nuclei are located the most closely to the basement membrane. Next will go the intercalated cells, and the most apical row of cell nuclei is the row containing cell nuclei of goblet cells and ciliated cells. So this is the um, cell nucleus of the ciliary cell. It reaches the basement membrane with its basal portion, and its apical portion reaches the surface of this epithelium, and it has cilia upon its surface. And as you can see here, the cell is filled with mitochondria. It needs a lot of mitochondria for movement of cilia. The goblet cells also reach the basement membrane, even if it is not seen in our particular pictures, but it is really so. So here is its nucleus, and the cytoplasm is filled with granules with mucus, which can be released to the surface, and this process is seen here. So function is mucociliary transport. Here you can see the dust particles which stick to the surface of epithelium, and this mucus with viruses and bacteria is moved with the help of beating of cilia. Below the level of the basement membrane, there is, as always, loose fibrous connective tissue with cell nuclei of cells of loose connective tissue and reddish eosinophilic, the collagen fibers. Another example of this epithelium. So basement membrane should be here. This is the basal cell. Next will be the layer of tall intercalated cells, and these are the cell nuclei of ciliated cells and goblet cells. Goblet cell is seen here, for example. It looks like a whitish bubble filled with mucus. Another picture is here. Once again, three rows of cell nuclei, basal, tall intercalated, and ciliary and goblet cells. Mucus with stick dust particles. So we have discussed the first part of the epithelium. This was the topic 5, if I'm not mistaken. And now we go to the second part of epithelia, the stratified and glandular epithelia. So once again, epithelia are divided into surface and glandular. We discussed this part of this diagram, surface simple epithelia. And now we will discuss surface stratified epithelia and glandular epithelia. So stratified epithelia include transitional epithelium, which is shown here in two pictures in non-stretched and stretched state. Number 2 is stratified squamous non-keratinized and stratified squamous keratinized epithelium. So in stratified epithelia, only basal layer of cells is located upon the basement membrane. All other cells have contacts only with other cells. Apical basal polarity here is the degree of differentiation of cells which corresponds to the location of the layer. So the stem cells will be located in basal layer, the cells in the process of maturation in the next layer, then mature, dying, aging, and dying cells finally at the top layer. Um, stratified squamous non-keratinized epithelium contains flattened living cells with cell nuclei flattened in its superficial layer. Stratified squamous keratinized epithelium has dead cells, squames with no cell nuclei in its top layer. And transitional epithelium has cells with rounded cell nuclei in its the most apical layer. So these are the main differences between these three types of epithelia. So you should look upon the surface every time. So here, all the three types of epithelium. First one is keratinized stratified squamous epithelium. It has 4 or 5 layers. First layer is called basal layer or stratum basal. The names will be mostly in Latin, so um, it may be hard for you to remember them, but try to. So, stratum basal, which means basal layer. One layer of cells lying upon basement membrane. Next layer is called the stratum spinosum. Stratum spinosum. The cells with different shape and mostly rounded cell nuclei. Stratum spinosum. Next goes stratum granulosum. It is located here, stratum granulosum. Granulosum is about granules. The cytoplasm of these cells is filled with small granules, so this is why it is called so. Next goes stratum lucidum. Lucidum means light looking, and the most superficial layer is stratum corneum. And this layer contains only dead squames, no living cells here. Next, we will discuss the non-keratinized stratified squamous epithelium. It has no stratum corneum, and you can see the living cells with cell nuclei flattened in its superficial layer. And it has only 3 layers. The most bottom layer is called basal layer once again. Next layer is stratum spinosum and superficial layer containing flattened cells. Stratum spinosum is the same as in the keratinized stratified squamous epithelium, also cells with different shape and rounded cell nuclei. Spinosum means small projections of the cells, which are seen in high magnification. We will see them later. And transitional epithelium also has 3 layers: basal layer, intermediate layer, and superficial layer. So, stratified keratinized squamous epithelium. Here in the picture, we can see the section of the skin of the finger. So, this epithelium makes the epidermis of the skin. The skin is the organ as the stomach or heart. So, as any organ does, the skin contains several types of tissues, and it contains the epithelial tissue in its top, and this area is called epidermis, the most superficial layer of the skin. So, what we should find first, we should find the border between the epithelium and underlying loose connective tissue. And at this border, the basement membrane will be located. So, here is this border. It goes like this. Here, number 7 is loose fibrous connective tissue. So here, number 1, basement membrane. Upon basement membrane, the basal layer is located. One layer of cells located upon basement membrane. If we look to this diagram of this epithelium, we can correspond the picture in the left and this. So basement membrane is the grayish line here. The basal layer or stratum basal is here. And in this layer we have dividing cells. So this is the cells during mitosis process. So stratum basal, basal layer. Next goes the stratum spinosum. All these cells are stratum spinosum cells. In the diagram they are here, cells with rounded cell nuclei and small projections which are called spines. So this is why it is called stratum spinosum with small spines. Next goes the stratum granulosum, number 4 here in this picture, and it is here, stratum granulosum. It is called so because of the granules. They are called keratohyaline granules, and they are mm, basophilic. They fill the cytoplasm of these cells. So this is stratum granulosum. Next goes the stratum lucidum, number 5. It is here. It is lighter stained stratum lucidum. Um, it is here. In this layer, the cells are dying and become post-cellular structures, the squames. And finally, the most outer superficial layer, number 6 here, is stratum corneum. In diagram, it is here. It consists of squames. One such structure is one squame. It is the post-cellular structure. It means that it previously was the living cells, but then the cell died and become the squame filled with the substance called soft keratin. Here, the process of keratinization is told in words, so you should read it yourself and try to mm, imagine how it happens and try to learn it. And I will try to explain this using the picture. So, the function of this epithelium is to protect the skin with the help of the thick stratum corneum. It is really very thick. If you look at the real picture of this epithelium, point to the surface, and most of the thickness of this epithelium, this is the stratum corneum, which consists of the dead cells filled with keratin. Keratin is the hard substance which also makes our nails and hair. So, what happens? Upon basement membrane, the cells of basal layer lie. Here they divide, they are stem cells, and the daughter cells start to differentiate. They next move upwards and become the cells of stratum spinosum with small spines. These small spines are small projections from one cell to another cell, and between those spines the spot desmosomes are made. So these all are spot desmosomes connecting the cells together. These cells have a lot of such desmosomes for more tighter, more harder, more strong attachment to each other because of the function of the skin covering our palms and fingers. This epithelium should be really very strong for us to grab something, etc. So in spines we have desmosomes. Desmosomes, as you remember, contain intermediate filaments. These are seen here. Intermediate filaments are made up from protein keratin. So the cells of stratum spinosum produce a lot of keratin protein and build intermediate filaments from this keratin. And these intermediate filaments um, are located around the cell nuclei here and also make the desmosomes. The intermediate filaments here are called tonofilaments. Next, when the cell becomes mature and next it ages and dies, it goes constantly upwards. So next, the tonofilaments, intermediate filaments, stick together to make more thick structures, which are called tonofibrils. Tonofibrils, they are located around cell nucleus. And next, they start to become glued together, they stick together with the help of the second type of protein, which is called filaggrin. And this combination of tonofibrils and filaggrin makes the so-called keratohyalin granules. So this is one granule, this is second granule, this is third, fourth, etc. They are rather large in size, so they are very easily seen in light microscope. So the keratohyalin granules, they are the granules which are seen in stratum granulosum. So these bluish granules are granules of keratohyalin which contain tonofibrils and filaggrin. Also, in the stratum spinosum, the second type of granules start to be formed, which is called keratinosomes. These small black circles, these are called keratinosomes. They contain lipids. And next, in the stratum granulosum layer, the keratinosomes go out via exocytosis, and lipids go between the cells and fill the extracellular environment between the cells. And then they will stick the squames together. Lipids are needed in the epithelium of the skin to prevent water and bacteria to enter our internal environment from the outer space through the epithelium to our blood and internal environment. So the surface layer of our skin has lipids between the squames. Lipids prevent water and bacteria to go through the epithelium. And lipids are produced inside keratinosomes. Keratinosomes are small granules not seen in light microscopy, only in electron one. What happened to keratohyalin granules with tonofibrils and filaggrin, which are basophilic and large? They become more and more compact, and they fill constantly all the internal space of the cell from within. Next, in stratum granulosum and stratum lucidum, the nucleus disappears, the lysis of organelles and nucleus happens, and the cell becomes gradually the postcellular structure, the squame. And this squame now is filled with the keratohyalin granules, which fuse together and make the substance called soft keratin. So the soft keratin actually contains tonofibrils bundles. So these lines are tonofibrils bundles, soft keratin. And the squames are connected together now, not with desmosomes like living cells are, but with lipids between the squames. And finally, the squames are exfoliated from the surface of our skin. So this is the process of the differentiation of the cells of this epithelium. The cells are called keratinocytes. They are called keratinocytes because they produce constantly protein keratin and become filled with the substance called soft keratin from inside. So their function is to produce and be filled with keratin, so they are called keratinocytes. In electron microscopy, this is how different layers of such epithelium look like. So here is the stratum basal lying upon basement membrane. Here is the basement membrane and the cells of stratum basal or basal layer. They are stem cells which have hemidesmosome to be in contact with the basement membrane. Next layer is the stratum spinosum, and you can see here these small spines connecting the cells together, and more darker structures here between the spines are spot desmosomes. So each pair of spines has a spot desmosome, and this is how it looks like in the light microscopy. So small projections are spines, and between the spines we have spot desmosomes for better attachment, mechanical attachment of cells together. Next layer is the stratum granulosum, in which the keratohyalin granules are located in cytoplasm of the cells. They are blackish here in stratum granulosum, and next layer will be stratum lucidum and stratum corneum. In stratum corneum, which is seen here, no cell nuclei and no organelles are seen, so these are squames filled with soft keratin from inside. In epidermis, which is the outer layer of skin as an organ, we have epithelium containing several cell types. The main cell type, or the main differon, is called the differon of keratinocytes. They are the major cells here, uh, all that are bluish in this diagram. All the bluish cells here are keratinocytes from the basal layer up to the superficial layer stratum corneum. All these are keratinocytes, but among these keratinocytes we can find also several other cell types, such as Langerhans cell. This one, it is the macrophage which performs the function of protection against bacteria. Some bacteria can still enter through the stratum corneum, and if it happens, the Langerhans cell will catch them, eat them, and protect us from bacteria and anything bad which can go through this epithelium. So Langerhans cell is for protection. Pigment cell or melanocyte has the pigment granules which gives our skin its color. Merkel cell is to feel touch, so it is the sensitive cell which feels the touch. So in the same picture that we have seen before, we can find all these cell differons. So most of the cells here are keratinocytes. All these are keratinocytes. Uh, what else can be found here? Langerhans cell, this one, macrophage, which catches bacteria. Next, pigment cell, melanocyte with granules of pigment, and Merkel cell. Merkel cell is for sensation of touch, and also lymphocyte is seen here. So this is the preparation of this type of epithelium, stratum keratinized squamous epithelium, in the so-called thick skin section. Our palms and feet have thick skin, and all other surface of our body is covered with thin skin. Those names, thick and thin, come after the thickness of stratum corneum. If the layer of stratum corneum with dead squamous is rather thick, then it is called thick skin. It is only in palms and feet. All other skin is thin skin. And second difference between those types of skin is the presence or absence of stratum lucidum. In thick skin, the stratum lucidum is present. It is the layer after stratum granulosum. Stratum granulosum is um, blue-purple here because of the keratohyalin granules in the cytoplasm of the cell. So above this layer there will be stratum lucidum. It is present here but absent in thin skin. So this is our epithelium. Below it we have loose fibrous connective tissue, more pale. So the basement membrane is located just at the border between the epithelium and loose fibrous connective tissue. One layer of cells standing upon basement membrane, they will be called stratum basal or basal layer. Next goes stratum spinosum from basal layer up to stratum granulosum. Stratum granulosum is readily seen because of the basophilic granules in cytoplasm. Next goes the stratum lucidum and the outermost stratum corneum. Embryonic source of development is ectoderm. Another picture. The same is here: stratum basal, stratum spinosum, stratum granulosum, next stratum lucidum, and stratum corneum. Another picture: stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, and all the rest stratum corneum. At high magnification, stratum basal, because below it we can see loose fibrous connective tissue, whitish. Stratum basal, stratum spinosum with pale cells. Next goes stratum granulosum, where we see the keratohyalin granules in cytoplasm of the cells. Next goes the stratum lucidum, and all the rest stratum corneum. The same as here. So this should be your picture in your album, task 31 if I'm not mistaken. So you should draw something like this with all the cell layers: stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. So as I uh, told you, we have thick skin in the palms and feet, and thin skin covering the rest of the body. So the main difference is the thickness of the stratum corneum. Compare the stratum corneum in left picture and stratum corneum in right picture. It is very thin, in thin skin. Um, Also in thick skin we have stratum lucidum, and we have no stratum lucidum in thin skin. In the preparation of thin skin, we also look at the sebaceous glands, which are located here. Sebaceous glands you have studied before. So in thin skin, what we have? We have stratum basal, number 2, located upon basement membrane, number 1. Below it we have loose connective tissue, number 6. After stratum basal we have stratum spinosum, number 3, thin stratum granulosum, basophilic, number 4, and stratum corneum, number 5. Go next to stratified non-keratinized squamous epithelium. It has 3 layers: basal layer lying upon basement membrane, next goes stratum spinosum, and superficial layer. There is no clear border between the stratum spinosum and superficial layer. We should look at the shape of cell nuclei. The superficial layer contains flattened cell nuclei. This is why this epithelium is called squamous. So where we see the flattened cell nuclei, so this will be the superficial layer. Where the cell nuclei are oval or round in shape, this will be stratum spinosum. So stratum basal, stratum spinosum, superficial layer with flattened cells. Here we have one cell differon, so this differon includes stem cells, cells in process of differentiation and mature cells, and then aging and dying cells. This epithelium we see in the section of esophagus. Here is this epithelium. This is the lumen of esophagus. The food goes here along the esophagus. Embryonic source of development of the epithelium of the esophagus is the precaudal plate, pointed with red arrow in both pictures, precaudal plate. So here is this epithelium. Once again, this epithelium, it has three layers: the basal layer, stratum basal, stratum spinosum, and superficial layer. Stratum basal cells look more dark basophilic because they have smaller volume of cytoplasm, and so their cell nuclei are located more closely together, and it gives us more dark basophilic look of this layer. In the stratum spinosum, mm, this layer looks more pale for us because the cell nuclei are located um, more far away from each other because the cells have larger volume of cytoplasm. And in superficial layer we have flattened cells. Um, as I said before, here in esophagus we also have glands, which we see here. We will talk about them later, but you should know that these glands produce mucus, which covers the surface of epithelium, because this mucus is produced here and is released via the ducts to the surface of epithelium inside the lumen of esophagus to moisture the surface of esophagus and to prevent friction and damage of the epithelium by food. And the glands which lead to the surface of some epithelium develop from the same embryonic surface as the epithelium with which they are connected with ducts. So these glands will develop also from precaudal plate as this lining epithelium does. So at high magnification, epithelium, stratified non-keratinized squamous stratum basal, stratum spinosum, superficial layer. These are mucus-producing glands, which produce mucus released to the surface of this epithelium here. Both structures, this epithelium and these glands, are composed of epithelial tissues, but this is the lining epithelium or surface epithelium, and this is glandular epithelium. Both are epithelial, and both have the same embryonic source of development, precaudal plate. Another example of such epithelium, this is not cross section of esophagus, but longitudinal section. So here is the same epithelium with stratum basal, stratum spinosum, and superficial layer. Another examples of the same preparation. And your task, where you should draw this epithelium with basement membrane, basal layer, stratum spinosum, and superficial layer of flattened cells with flattened cell nuclei. Next, transitional epithelium. Transitional is called so because epithelium changes when epithelium gets stretched. So when the epithelium is distended, stretched, the overall thickness of epithelium decreases if we compare um, the picture in the left part with this one. Also, the shape of the cells of the superficial layer changes. Here, in a stretched condition, they are stretched. And also, number of rows of the cells in intermediate layer number 3 changes. When the epithelium gets stretched, some of the cells get inserted between the cells of the basal layer because they are actually connected with basement membrane. So in non-stretched condition, epithelium looks like here. 1, 2, 3, 4, or 5 rows of cell nuclei are seen here, but only 1, 2, 3 rows of cell nuclei in stretched condition. Also, overall thickness of epithelium decreases in stretched condition, and also the shape of the superficial cell layer uh, changes. So in superficial layer, cells become more flattened. Here, this epithelium we see in the section of urinary bladder. This is the lumen of urinary bladder, which should be filled with urine. So this is the epithelium of the urinary bladder. Its embryonic source of development is endoderm, partly the embryonic endoderm and partly the extraembryonic endoderm of allantois, which is here. In the same picture, we can find the part of epithelium which is stretched and part of the epithelium which is not stretched, where we have the fold. So we can compare these areas, overall thickness in stretched and non-stretched, the number of cell rows, here 3 and here 1, 2, 3, 4 at least in non-stretched, and the shape of superficial layer, more flattened in stretched area and more dome shaped like this, rounded, in non-stretched area. The same as here. But always the shape of cell nuclei in the superficial layer is round, unlike stratified squamous, keratinized and non-keratinized epithelia, where we have flattened cell nuclei in the superficial layers. What else we should know about the cells in this epithelium, which is also called urothelium, is that the cells of superficial layer have very specific apical cell surface specialization. This specialization is called the plates made up of uroplakin protein. So what is shown here is the cell membrane, and the outer layer of cytoplasm, the terminal web with elements of cytoskeleton connected to the cell membrane. And the cell membrane contains phospholipids and transmembrane proteins. And these cells have very specific transmembrane proteins, which are called uroplakins. This is how they look like if we see them from the top. They are combined together to form such plates located between the phospholipids. So the areas between these plates are phospholipids, areas which are flexible areas of the membrane. And the plates made up of uroplakin proteins are not flexible, they are hard. What is this for? This is for changing of shape of the cells of the superficial layer of this epithelium. So in stretched area, the membrane looks like this when the cells are stretched, but when the cells are not stretched, uh, the membrane of the apical surface of the cell is partly infolding inside of the cytoplasm of the cells. So the part of membrane makes the infoldings, and the surface area of the apical surface of the cell decreases when the epithelium is not stretched. but when it gets stretched once again, these folds go out and the apical surface gets stretched. So this is one of the functions of the uroplakin plates. So it provides stretching of the cells of superficial layer during distension of epithelium, and it has a second important function. It prevents water passing through membrane from the cell into the lumen of the urinary bladder. Why so? Uh, because here will be the lumen of the mm, urinary bladder filled with urine. Urine is more concentrated fluid than the fluid filling the cell cytoplasm. It means that water will go out from the cell to more concentrated fluid, to urine. And if this happens, then the cell loses water and dies. To prevent this, the cell has these specific plates which prevent passage of water through the cell membrane. So water cannot go from the cytoplasm of the cell towards the urine, which fills the lumen of the urinary bladder. So this is the second function of the uroplakin um, protein. And now we go to glandular epithelia. Glandular epithelia make glands, which produce different types of secretion. Uh, Glandular epithelia have some different classifications according to the chemical composition of the secrets. We have the protein glandular epithelium, or serous is the same. Such cell which produces proteins has a rough endoplasmic reticulum, which makes the cytoplasm basophilic, so this is why the cytoplasm is basophilic. Golgi complex should be here, non-stained, and secretory granules filled with proteins, which usually are stained eosinophilic. One example of this serous gland. Next, mucus producing gland or mucus producing glandular cell. This is such a cell. It has flattened cell nucleus located squeezed closely to the basal portion of the cell. Here should be the basal surface of the cell with basement membrane below it. Why the nucleus is flattened and moved to the basal portion? Because it is moved with the vesicles filling all the cell, and the vesicles are filled with mucus. They are non-stained, so this cell stays non-stained, pale, and this is how this cell looks like goblet cell or other type of mucus-producing cells. Next, water and salt producing glandular cell. It also has almost non-stained, pale stained cytoplasm because um, it has low amount of different organelles, and you can see here such cells in the section of sweat glands. And here lipid or steroid producing cells. So these cells have pale cytoplasm because it will be filled with smooth endoplasmic reticulum, which is for lipid production, and um, secretory granules, vesicles filled with lipids. Lipids are also not stained with um, either eosin or hematoxylin, so these vesicles are also non-stained. So such cell also will be with pale cytoplasm. So here you can see such cells. Only their nuclei are stained, but cytoplasm is not stained. It stays pale. The examples of such cells: protein producing cell is, for example, located in salivary glands, which produce saliva in our mouth. Saliva has in its composition some proteins such as antibacterial proteins. proteins such as lysozyme and lactoferrin. They are constantly released um, with the help of these serocytes, the cells producing proteins, and these antibacterial proteins are constantly located inside our um, mouth cavity to fight bacteria. Mucus producing cells are also the part of salivary glands. They produce mucus component of the saliva. Water and salt producing glands, examples are sweat glands in our skin, which produce sweat, and also lacrimal glands, which produce tears. Lipid producing glands, they are sebaceous glands in our skin, which produce lipids, sebum, and steroid producing glands, they are, for example, adrenal cortex, the cortex of adrenal gland, which produces steroid hormones. Um, Glandular epithelium cells has its secretory cycle, which starts with the uptake of chemical precursors. For example, this cell produces proteins. Why do we think so? Because it has a lot of rough endoplasmic reticulum here. Its cytoplasm is filled mostly of rough endoplasmic reticulum. It has Golgi complex and dark-looking vesicles because they contain proteins. So um, the precursor for proteins is amino acid. So this cell takes amino acids which go via diffusion from the blood capillaries through basement membrane to this cell. So it takes amino acids. Amino acids go to rough endoplasmic reticulum where the proteins are synthesized. So second step is synthesis of the product. Then this product with the help of vesicles go to Golgi complex where it is changed somehow and packed into vesicles. So here's the accumulation and maturation of the product in Golgi complex, and then the vesicles containing proteins are accumulating in the apical surface of the cell um, up to the signal for a cell to release these granules. So the last stage will be releasing of mature product from the cell. Um, so the stage of the synthesis and accumulation takes about 1 hour, and other uh, stages take a few seconds up to a few minutes. Glands are also divided into exocrine and endocrine glands. Um, so, uh, exocrine glands release their secrets um, out to the surface of our body or into the lumen of organs. For example, uh, the goblet cell is the exocrine gland which releases the mucus into the lumen of intestine. And here we have um, the cells in pancreas, which are also exocrine gland cells, which release their substances, their enzymes, into the lumen of the uh, duct of the pancreas and finally into the lumen of the duodenum intestine. Endocrine glands release their substances into blood. Exocrine glands are subdivided into unicellular or endoepithelial and multicellular or exoepithelial. Uni means one, cellular means cells. So unicellular means containing one cell. The gland, which is actually one cell. So this unicellular gland is, for example, a goblet cell. So it is glandular cell inside the epithelium, inside the lining epithelium. And so this is why it is called endoepithelial gland, because it is located inside the epithelium. Endo means inside. Multicellular means many cells in the structure of such gland, and it is located outside the epithelium. Um, Here we have the example of the duodenum. This is the first part of the intestine, which goes just after the stomach. And here you can see the villi, the crypts, lined with this epithelium with brush border. And also here we have the glands located lower. And these glands here, we can see their structure. They are multicellular because they have many cells in their structures. They have the part which produces secrets, which is called secretory portion, and the duct, the tube which leads to the surface of epithelium, through which the secrets goes out to the surface. So these are secretory portions, and the ducts will lead to the surface of this epithelium here. These glands will produce also mucus in this particular example of duodenum. And the exocrine ducts in embryogenesis develop from the epithelium, with which they are which they stay connected with their duct when they are fully grown. So, unicellular gland is the goblet cell, gland containing only one cell, and multicellular gland, which contains many cells, such as the acinus of the pancreas. Acinus is the name of the secretory portion, the combination of cells which produce and release secretions. Um, Simple columnar epithelium with brush border and goblet cells inside it. So this is unicellular endoepithelium mucous exocrine gland. So it is the full name of this cell. Exocrine and endocrine glands. So exocrine glands have ducts. So this bluish area is the duct, the tube through which the secrets go from secretory portions where they are produced. Secretory portion here is yellowish. So ducts, exocrine glands release their secrets outside the body or into the lumen of an organ. But endocrine gland has no duct because it releases secrets which goes directly to blood capillaries.
اینو نمیگم فیکس پرایس از سنجاقایی که
So, for example, in pancreas we have both types of glands. We have exocrine glands, this part which has secretory portion with glandular epithelium, and ducts, which are tubes lined with surface or lining epithelium. And also here we have the endocrine portion, which is endocrine gland, made up with glandular epithelium. These cells produce some hormones such as insulin. So our pancreas is the combination of exocrine and endocrine gland, which produces enzymes for digestion in intestines, and enzymes are produced by exocrine portion of this gland, and also hormones such as insulin, which are produced by endocrine portion of this gland. These hormones go directly to blood capillaries, which are seen here cross-sected.
همینجوری واضح عکس میده
So this is the intestinal crypt. What is crypt? Once again, crypt is located here. So these are crypts. They are invaginations of epithelium long. And here you can see the epithelium which lines the crypts. It lies upon the basement membrane. It is located here. Below
خیلی چرت و پرت
basement membrane there is loose connective tissue here, and we can find here exocrine and endocrine endoepithelial glands. Means
Tamam
that
ya mesela
they
اینجا
are
باید از روش
just
ببینیم چه
cells
توضیحشون داده
which produce some
اینجا میگه
substances and release
پلیزوم
those
اگر میگه
substances
اینجا نشون میده میگه
either
چیه
outside
باید بگی
into the
پلیزوم
lumen
چون
of
چند
the
تا چیز
uh,
میشن نوکلئوس میشن
intestine
اینجا
or
شبیه
into
آژینا
the
هست چون
blood vessels,
معلومه این
into
توضیح
blood
خیلی
capillaries.
معلومه چی داشت
So, mm, goblet cell is the unicellular endoepithelium exocrine mucus producing gland. It is exocrine because it releases substances into the lumen of the intestine. And also, we can find here other type of exocrine cells, which are called Paneth cells. They release also antibacterial proteins, and they are seen here inside the uh, vesicles. So, if we look at the cell, how it looks like, we can say if it is an endocrine or exocrine gland, according to the location of the secretory vesicles. If the secretory vesicles are located in the apical portion of the cell above the nucleus here, it means that they will be released through the apical surface into the lumen of the organ. And it means that this is the exocrine gland. But if we look at the cell here, it has the secretory vesicles in its basal portion below the nucleus here, and it means that these vesicles will be released through the basal surface of the cell, and the content of these vesicles will go through basement membrane to underlying connective tissue and finally to blood capillaries. And it means that we see here endocrine cell. So we should look at the location of secretory vesicles. So here in the picture in the left we have the exocrine cell because it has secretory granules, secretory vesicles in its apical surface. Why it is apical? Because below the nucleus we have a lot of granular rough endoplasmic reticulum, and here we have basement membrane. Where the basement membrane is, there the basal portion of the cell is. And here we have the duct of the gland, so here should be the apical portion of the cell. So we can say that this cell is exocrine cell. And if we look at this cell in this picture, we can say that here is its apical surface because here it has the microvilli, which are the specialization of apical surface, and here should be basement membrane, so here should be basal surface, and the cell has secretory vesicles, dark black, in its basal portion. It means that this is endocrine cell. It will release those granules through basal portion, through basement membrane into blood capillaries, like this. And if we look at this cell, it also has secretory vesicles in its basal portion, but what are the difference between the cell below here and in the upper picture? Um, in the picture below, our endocrine cell reaches the surface of the epithelium, but in the picture above, our endocrine cell doesn't reach the surface of epithelium. So in this case, this endocrine cell is called the endocrine cell of closed type, and in this case, this endocrine cell is called endocrine cell of opened type because it reaches the surface of epithelium. Now, classification of glands according to modes of secretion, of how the cell releases its secretory vesicles. And we have merocrine, apocrine, and holocrine glands. What does it mean? Merocrine glands release their secretory vesicles via just ordinary exocytosis. It means that a secretory vesicle membrane fuses with the cell membrane and the content goes outside. Um, in apocrine glands, the vesicles are accumulating in the apical portion of the cell, and when it is needed, all this apical part of the cell detaches from the cell and goes out. And it means that in this case, the apical portion of the cell containing secretory vesicles is broken down via the process of the release of secretion, like is shown here. And in holocrine type of secretion, the cell is gradually filled with the secretion, then nucleus and organelles degenerate, and finally all the cells is broken down and become the secretion itself. The example of the holocrine type gland is the sebaceous gland, which produces sebum, lipids in our skin. Next, classification of glands according to their morphology. This is the classification for multicellular exocrine glands. These glands have um, secretory portions. Secretory portions here, where we have glandular epithelium, the cells which produce secretion, and duct, the tube through which this secretion goes out. So, this classification based upon the morphology of secretory portions and ducts. So, um, first, according to the mm, branching of duct, we have simple or compound glands. If the duct is not branched, the gland will be called simple. If the duct is branched, then the gland will be called compound. Next, uh, according to the branching of the secretory portion, we have branched or unbranched gland. And finally, according to the shape of the secretory portion, we have tubular, alveolar, or tubular alveolar gland. Uh, what does it mean? It means that based upon their classification, and you should be able to give full name for any gland, the diagram of which you see. Uh, how it works? Look at this picture. Um, this whitish area, lighter, this is the duct, and dark black, this is the secretory portion. So this will be simple, non-branched tubular gland. First name, simple, goes after the branching or not branching of the duct. Duct here is not branched, so the first name of this will be simple. Next, we look at the secretory portion, if it is branched or not. It is not branched, so the second word in the name will be unbranched. And finally, we look at the shape of this secretory portion. It is like a tubule, so it is tubular, and the word will be tubular. So, a full name for this gland will be simple, non-branched tubular gland. Go next here, the same. Duct is not branched, so the first word will be simple. Next, secretory portion also is not branched, so it will be non-branched. And the shape of the secretory portion is alveolar. Alveolar means like a bubble or vesicle, like this, alveolar. So the name will be simple, non-branched alveolar gland. Now go here. Here we see that duct is not branched, so it will be simple, but secretory portions are branched, or not one but few secretory portions lead to one duct. It means that the second word in the name will be branched. And then third word will be about the shape of the secretory portion. Again, it is tubular, like here. So this gland will be called simple branched tubular. Next here, this will be simple branched alveolar. And here you can see that both duct is branched and secretory portions are branched. More than one secretory portion opens into one duct, and also we have different shape of secretory portions. So first word will be compound, not simple. So simple compound. According to the branching of duct, here we have compound, branched, tubular alveolar gland. So here, number one, simple non-branched tubular. Number two, simple non-branched tubular with twisted secretory portion. This is the diagram of the sweat gland. Sweat gland looks like this. Next, number three, simple branched tubular. Number four, compound branched tubular. Number five, simple non-branched alveolar. Number six, simple branched alveolar. Number seven, compound branched alveolar. Number eight, compound branched tubular alveolar. So, um, if we use the preparation of the section of our skin as an example, there in skin we will see two kinds of glands. Sebaceous gland, which produces sebum, lipids, here we see it, and sweat gland. Sweat gland is here. Sweat gland produces sweat. Both release their secretions to the surface of the epithelium of the epidermis, so they develop from the same embryonic source of development, from ectoderm, and their shape is described as the follows. So the shape of the sebaceous gland is simple branched alveolar, like number 6 in here. Simple branched alveolar, non-branched duct, and two or three secretory portions lead to the duct, and the shape of secretory portions is rounded in shape. And sweat gland, here you can see the section of sweat gland. Its shape is like in number 2, simple non-branched with twisted secretory portion, tubular. So this is the duct, and here you can see the sections of both ducts and secretory portions, because it's the continuous tube which is very twisted, and when we make the cut through this twisted secretory portion, we will see a group of ovals and circles in one area. Some of these circles are secretory portions, some of them are ducts. We should look at the size and color of the cells. The cells of secretory portions are usually larger and lighter than the cells of ducts, because the secretory portions are made up with the glandular epithelium cells, which have, like we see here, higher amount of cytoplasm because of a lot of organelles needed to produce some secretions. But the ducts are made up with the lining type of epithelium with a smaller amount of cytoplasm, because they do not need to produce anything, they just provide the secretion from secretory portion outside. So this is why secretory portions look more pale and large than the sections of ducts. So here you can see the full description and classification of glands that we see in our classes and in colloquium, so you should study this slide for yourself. And here you can see also the classification according to the terminal portion, cell type, and um, the shape of the cells, and which organelles are developed in such cells. For example, if we have the serous secretory portion, there will be seen serocytes which have columnar or cuboidal shape with basophilic cytoplasm because of a lot of granular or rough endoplasmic reticulum, um, not very well seen lumen and borders between the cells, etc. But in mucus producing cells, we have um, very light cytoplasm um, with clearly visible borders between the cells and the lumen, and the dark flattened against the base of the cell nucleus. So we will see different types of cells in the section of the tongue. So it's this section of a tongue inside which we have the salivary glands. So this is the surface of the tongue covered with stratified non-keratinized squamous epithelium. Below it we have loose fibrous connective tissue. Next goes skeletal muscle, which makes our tongue motile. We can um, move our tongue as we want because we have skeletal muscles here with muscle fibers. And between muscles, we have here groups of secretory portions of salivary glands. These are secretory portions, and these are ducts which lead to the surface of epithelium. Each secretory portion is connected to duct, and duct is connected to surface of epithelium, but it is not usually seen in our picture because of the plane of section. For example, here you can see how this duct leads to the surface of epithelium, but here you do not see it because of the plane of sectioning. But we know that ducts should lead to surface of epithelium. So here we have mucous and serous glands. At high magnification, how they look like. These are secretory portions of glands. They are made up with glandular cells in serous secretory portions. Serous means protein producing. The cells are called serocytes. They produce proteins, the proteins of saliva, such as antibacterial proteins, lysozyme and lactoferrin, against bacteria. So they are proteins, and they are made up inside rough endoplasmic reticulum. Rough endoplasmic reticulum makes the cytoplasm basophilic. The nucleus is rounded in shape, and the lumen of the secretory portion is not very well seen. It is here, lumen of secretory portion. Another second type of cell in secretory portion is the so-called myoepithelial cell. They are rather small cells here, located upon the same basement membrane. They have long processes that hug the secretory portion from outside, and they are called myoepithelial because they somewhat resemble muscle cells because they have the contractile myofilaments inside their cytoplasm, such as actin and myosin microfilaments, that help them to contract their processes, and when it happens, their processes squeeze the secretory portion, and the secretory granules are released from the cells through the lumens of the secretory portions towards the ducts. So the function of myoepithelial cells is to help release of the secretion from the secretory portions towards the ducts. Myoepithelial cells are present here in all the types of secretory portions. We have here three types: serous, mucous, and mixed. Serous we have discussed now. So these are the cell nuclei of serocytes, and myoepithelial cells have flattened cell nuclei like this. Next, mucous secretory portions. They are made up with mucocytes or mucous cells. They have pale cytoplasm filled with secretory granules with mucus, which is not stained, like in goblet cells, and flattened cell nuclei. So this is how such secretory, secretory portions look like. This is the lumen of this secretory portion, and these are the cells. You can easily see the borders between the cells, and their nuclei are here, closely to the basal surface of the cells. They are flattened like this. So cell nuclei are here, and also the myoepithelial cells are present somewhere also here. They produce mucus. And finally, we have mixed type of secretory portions in which we have both cell types, mucus producing and serous producing cells. So this is how such secretory portion looks like. It has mucocytes with pale cytoplasm and flattened cell nuclei, and serocytes, protein producing cells, located at one end in hemilunes, such as crest, moon crest, in the form of moon crest. Here they are. They produce proteins, and also some myoepithelial cells can be also seen because of their flattened cell nuclei. So in our histological preparation, what we see: surface epithelium, underlying connective tissue, and glands. The mucous secretory portions, they are mucous because their cytoplasm is pale, non-stained. Serous secretory portions because of the basophilic cytoplasm of the cells. And here we have the ducts. Secretory portions are made up with glandular epithelium, and ducts are made up with lining epithelium. Embryonic source of development is ectoderm, the same as for the epithelium, which covers the surface of the tongue. Here you can see the group of sected secretory portions of the mucous gland because it is pale in color, and here you can see the duct leading to the surface of this epithelium. Again, here serous secretory portion with serocytes, protein producing cells, and mucous secretory portion made of mucocytes because of pale cytoplasm and flattened nuclei. Another example of serocytes. So this is one secretory portion. For example, here another one secretory portion. Secretory portion contains serocytes. Here you can see their cell nuclei. The lumen is here. The secretion will be released here, and also a flattened nuclei of the myoepithelial cells. At high magnification, serocytes, not very well seen borders between the cells, basophilic cytoplasm, round nuclei. These are the features of protein-producing cells. And this is the flattened nucleus of myoepithelial cell. These are mucus-producing cells in mucous secretory portions. This is mucous secretory portion with its lumen, cells with pale non-stained cytoplasm and flattened nuclei. These are mucocytes, and also some myoepithelial cells. At high magnification, you can see one and two secretory portions, mucus-producing secretory portions with mucocytes. You can see the borders between mucocytes. There are cell nuclei flattened, squeezed to the basal portion of the cell. Here is the lumen of the secretory portion. Another example. And we go next to next preparation. Here you can see the section of the uterus of a cat. Uterus is the organ where um, baby grows during pregnancy in women. So this is the cross section of uterus. Uterus also has the cavity which is called lumen. It is here, lumen of the uterus. It is not round in shape. It is like a star because it doesn't stay always open. So it's the normal shape of the lumen of the uterus. Um, Lumen of the uterus is lined with simple columnar epithelium, which you can see here, and also the glands are present here. The glands are simple tubular. Embryonic source of development is nephrogonotome. As you remember, nephro is for urinary system, gono for gonads for reproductive system. So at high magnification, we can see here the lumen of the uterus lined with simple columnar epithelium of lining or surface type, and the glands. Glands are simple tubular. Simple, it means that the duct doesn't branch, and non-branched tubular is about the secretory portion. This gland has parts which are isthmus, or the opening of this gland, opening to the cavity. Next goes the neck of the gland. Next, body of the gland. Most of the part, most of the length of this gland is body, and the bottom. Um, Here we have the lumen, cavity of this gland. In this lumen, the secretion released by the cells, and then this secretion goes through neck, through isthmus, to the lumen of uterus. Gland is made up with glandular epithelium, and the neck and isthmus and lining epithelium here is lining or surface epithelium. Another example of the same preparation, lumen of the uterus, lining epithelium of the uterus, and glands. Um, we not always see how the uh, lumen of the gland is interconnected with the lumen of the uterus, but this is because of the plane of section. If we look here, for example, it may seem that this gland is not connected to the lumen, but it really does, but not in this section. So here should be the isthmus, neck, body, and bottom of the gland. What is important thing about the bottom of the gland, that in this particular glands, the stem or Campbell cells are located exactly in the bottom of the gland. And this is very important because of the menstrual cycle in women. And during this menstrual cycle, all the internal part of the uterus dies and is shed away from the body of woman with bloody fluid, and only the bottoms of the glands stay inside the body of woman, and from these stem cells new epithelial cells are regenerated, making new glands and new epithelium which lines the cavity of the gland of the uterus. So in your picture, what should you draw here in task 34? You should draw one gland here, tubular, and make these labels. First, the lumen of the uterus, the gland with its parts, isthmus, neck, body, and bottom. Basement membrane is here, and below the basement membrane, outside the basement membrane, there is loose fibrous connective tissue. Next preparation is simple branched alveolar gland, the sebaceous gland of the skin. So this preparation is the section of the thin skin containing hair. So these are hair, which we see here. This is the surface of epithelium. This is epidermis, which contains stratified squamous keratinized epithelium. And we should find here the sebaceous glands. Sebaceous glands are connected with their ducts to the uh, structures of hair. So where the hair is, there the sebaceous glands should also be found. So if we trace one hair from its bulb, where the stem cells for hair are located, we trace it to the surface of the skin here. Closer to the surface, we will find here the sebaceous glands. Here we can see their secretory portions. Another example of this preparation, the epithelium, which is stratified keratinized epithelium, the hair, it is seen here, and sebaceous gland, one here and another one here. In this area, you can see also the sweat gland, which is very twisted. Here, uh, at high magnification, you can see the structure of the sebaceous gland. Sebaceous gland, it is simple branched alveolar. Simple because its duct doesn't branch. Next word, branched, is about the secretory portions. Um, not one but two or three secretory portions can open into one duct, but it is not seen in this particular picture. In this particular picture, we can see only one secretory portion, which is lined with this dashed red line here. This is the secretory portion. And the secretory portion is the vesicle, bubble looking, so it is alveolar. Um, so secretory portion contains cells which are called serocytes, and um, you know about serocytes that they have the Campbell cells or basal cells here at the periphery. Next, we will have cells in the process of differentiation, mature cells, then aging and dying cells, and finally, dead and broken cells containing lipids are released through ducts. So, here we can see the Campbell cells, mature cells, aging and dying cells. All of them are sebocytes. Inside the cytoplasm, we can find the lipid droplets. They are not seen, so look like they look like white bubbles inside the cytoplasm. And also, the cells will have smooth endoplasmic reticulum to produce a lot of lipids. So, in your task 36, you should draw such a gland with all the labels. So, this is all. If you have any questions, I will be glad to answer them. If not, I wish you a good day. So, goodbye everyone. Wish you a good day.